Good. Okay, so the first talk I wanted to give for our online session is kind of a blend of techniques from biology and the field of bioinformatics, computer science, statistics, and some, kind of modern interdisciplinary ideas, but all to solve a very relatable and, important problem, understanding cancer more deeply. So first I want to just summarize the big picture of what this paper is doing. So you can take a look at the paper afterwards if you like. I have it in this other tab, and I've also included it in the email. But just the big picture of what they're trying to do here is to look at a group of patients, all of whom have breast cancer, and they're trying to find a way of predicting, given a patient, whether their breast cancer has metastasized. So before I start throwing around words, does anyone, especially the students, can anyone define for me what the word metastasize or metastasis is, if you've heard it before? Does anyone know? I'm just going to draw a lot of question marks.
Is it vaguely related to metaphase?
No, okay, so metastasis is kind of an organism level phenomenon, not a cellular level phenomenon. So you probably haven't heard of it in biology. You might have heard about this if you've been reading about cancer kind of on your own, or if you've just heard it. Okay, Adam, do you know what it is?
When something metastasizes, I am under the impression that means that's when it becomes a malignant, right? Where it starts growing out of. That may be wrong. I'm not sure.
Ah, good. So it's related in the sense that metastasis is part of the progression of tumor through malignancy, but specifically metastasis is when the primary tumor, so if you first get one type of cancer, say in the colon, metastasis is when those cancer cells from the first tumor break off and start to make new tumors elsewhere in the body. So this is after the malignancy. Malignancy is a first step where the primary tumor becomes malignant and then it spreads elsewhere, right? So metastasis is another word for effectively spreading. Right. So back to the big picture, this metastasis is something very important to predict, because if someone has breast cancer, they normally don't die from the breast cancer itself. That primary tumor is not going to be fatal, but metastasis to other important organs like the brain or lungs or heart is what most often kills the patient. And it's very challenging to tell in the early stages of metastasis whether it's happened, because you can't just look for very small tumors everywhere in the body. So what researchers would like is a way of taking in a so-called expression profile, which is basically lab results. You take a bit of the tumor and send it to the lab and do some tests on it and get an expression profile for proteins. Then what we would like is a box, some sort of predictive model that you feed the lab results into this box, and this tells you a yes or no. Do we think that this primary tumor has metastasized somewhere else? Right. So to be clear, we want to be able to look at the primary tumor and be able to guess whether it's metastasized without looking anywhere else in the body. So this is the problem we want to solve, the goal in this paper. The primary tool that the researchers use in this paper is something called a protein-protein interaction network, which is a sort of mathematical model. So we'll have to go through in a bit more detail at this point. I'm throwing words at you, but the goal for the rest of the talk is really to understand how cancer and metastasis work, what these protein networks I'm talking about are, and then how they help solve the problem. I should look to see if anyone else is pinging. Corbin's being flaky. Okay, good. Okay, so my plan is going to be to first give a big picture summary of the paper. Here's a simplified flowchart of what they do. Then I'll go through and try to explain some of these steps in more detail, and then we can do the flowchart again with it unsimplified. Good. So effectively what they do is use this idea of a protein-protein interaction network, which is this structure where the individual dots are proteins. We call these nodes. Each dot is a different protein, and a connection between nodes is an interaction. So they have this structure. I'll tell you where they get it from, but they have this idea of these are how the proteins interacting. And they have some profiles. So this is what we would call a training set. These are patients that have breast cancer that has not metastasized in blue, and which have metastasized in pink, and their lab results from these guys. So then what they do in the paper is to take the protein network they have and look at various sub-networks, so smaller parts of the network, and then they look at the data from their patients. So they collect all of this data into a table where the individual rows are different proteins in the network, and the columns are different patients that are either metastatic or not metastatic. And then they try to compute some number which measures whether each of these subnetworks has a different amount of protein for the proteins in the network, a different amount of protein between the two samples, between the metastatic and the not metastatic patient types. And then the output is they're going to find, okay, these are the networks which significantly differ. These are clusters of proteins that have different levels in the two types, the metastatic and non-metastatic pools. So that's what they're trying to do. Find these guys that will tell you whether a patient is metastatic or not metastatic. That's what they do. I'm going to jump right to the punchline to summarize what they found before going in greater detail. But the sort of prediction, that box they get, this model at the end of the day they get from this procedure, has better accuracy at predicting whether someone is metastatic. They beat this baseline. Some guys got a 63% prediction accuracy using only individual proteins. These guys get 72% by using the network. So that's here's a different measurement. This is the area under the ROC curve, which is somewhat less important, but it's something which tells you how good is your prediction algorithm. And this is what these guys got, these subnet markers. So they beat the existing techniques by a pretty substantial margin. And there's other things that were good about this. Consistency between patients and generating novel hypotheses and so forth. So they had very nice results, is the point. And this paper is highly cited, which is why I chose it. So this is like a rapid intro before going in somewhat greater detail. But so what we'll do next, now that we have sort of some situation of what we want to do, first I want to do basically a review of, 10th grade bios. This is, whatever Mr. E or Mr. Miller or whoever you had for bio has probably told you a lot of this stuff, but I'll review it for you quickly. And I'll tell you how you tell whether proteins interact. Then I'll tell you more about these networks, these protein-protein interactions. Then we'll talk a little bit about statistics, and then I'll come back and redo that intro that I just did with a little more detail now that we understand the pieces. So good, let me pause here. Before doing Act 1, thoughts, questions, comments, or death threats from anyone?
It may be worthwhile to mention your usual phrase of not needing to understand everything that is being said during a presentation.
Yeah, good. So I considered bringing this up separately, but I didn't want to scare people. But yeah, I think when you listen to a high school class, like a lecture or something, you usually expect to understand, maybe 60 to 90% of what is said. Everything is said at a level that you're expected to know what they're talking about. For a scientific talk about a paper, the threshold is much, much lower. You might, in a talk like this, expect to understand 10 to 30% of what I'm saying. And a lot of people get turned off by this and say, oh, I don't know what's going on, I'm going to leave. And they run away. That's the wrong attitude. So I think it's better to accept a lot of this is not going to make sense. And it'll be tempting to disengage and sort of not listen or zone out. But it's really quite good practice to stay focused anyway and try to black box and say, even though I don't understand all these details, can I try to extract what the main idea is of what's being said? I'm sure Adam can attest that most of the research level talks that you listen to require that skill, at least for me.
Yes, absolutely. Yeah, just kind of speaking to that, even having spent a few years in a field now, I regularly go to seminars where I get maybe 20 to 30% of what's being said. So yeah, this is a good skill to practice. I second that very strongly.
Good. Thank you, Logan. Good.
One quick question: when was this paper written again, or when was this published?
Good. So this breast cancer one. Let me go to the top. This is from 2007. In fact, this is already somewhat.
Okay, cool.
Yeah, I included two papers because this protein network stuff is quite new. One of the two seminal papers that basically started the whole field was this yeast one, which is actually from 2000. So they basically catalyzed this whole new technique, and that was only about 20 years ago. And then this one sort of expanded on it in 2007, and now there's just a lot. Both of these have thousands of citations already.
Interesting. Was the yeast one that 63% accuracy model?
Oh, no. So surprisingly, the number 63 appears in the abstract, but it's not the same 63.
That's what I was jumping off of.
Good, good. Yeah, this had nothing to do with breast cancer. They were just saying, like, can we construct a network for all of the proteins in yeast? And it's like a big, ambitious task. Yeah. Good. More questions? Or thoughts or objections? Or anger?
I do not have any thoughts.
I feel like it'd be quite concerning if somebody had a death threat about that.
I rarely get death threats. I always ask for them, but people never indulge me in the death threats.
I don't really think that that's something that you should really be asking for, but I mean, I guess.
We're not within our rights to stop him.
Yes. Okay, I'm very masochistic. I want people to want me to die or something. I don't know.
Question mark. Okay.
Very common at a seminar.
To have.
Oh wait, sorry, I missed what you said. What was it?
Was it just
I don't really think that's a very healthy trait to have.
Wanting to die? Yeah, probably not. Well, not wanting to die. Wanting other people to want me to die.
Yeah, I don't think that's how that's supposed to work. But I mean, like I said, whatever floats your boat, I guess.
Well, good. If death threats occur, you can shout them out at me. But
Right, if anything comes to mind, I'll be sure to shout it out impulsively.
Good. Okay, so let's do the first part, which is mostly stuff you would see in like 10th grade bio with Mr. E, possibly with different words. So I'll ask you if anyone remembers. Does anyone remember this term, central dogma of molecular biology, or just central dogma? They may not have used that word in bio class here, but they certainly use this, say, on the bio SAT or whatever. So I'll give you a hint. This is a relationship, sort of a flowchart between DNA, RNA, and protein. So can anyone tell me what that is, or how those three things are related?
Transcription, translation. That's all I gotta say.
Yeah, good. So which two are related by transcription, and which are related by translation?
I believe, RNA and I want to say RNA and protein are transcription or translation.
That one's translation. So RNA is translated to protein. By the way, can everyone see the screen share, or is the quality like sucky?
I can see it.
Good.
Yeah, I'm fine.
Good. Yeah, indeed. I don't know why I wrote translation instead of translation, but indeed.
Now that might need a death threat.
Good. Yeah, I got my first one. Good. Yeah. So this is I like to phrase it: DNA makes RNA makes protein. Right. So this is the sort of flow of information in the cell. The DNA is sort of the place where this is stored, the top level, and then as needed, DNA is transcribed into messenger RNA, which then is also translated into protein, and this is sort of the bottom. So why am I telling you about this as a review? Well, we're going to try to understand more closely what cancer is, which is really a disease of DNA, far at the left. And then I'll explain that because measuring DNA directly is hard, we can use this flow to see how changes in DNA might have occurred just by looking at the end result, the protein. So that's going to be the idea. First, let me tell you what cancer really is. So cancer is not a single problem, a disease with a single cause. It's a disease of time and accumulation. Right, so cancer is the end result when you have many small changes in cellular DNA, called mutations, as you know, over a long period of time, which affect protein production. And in particular, those mutations which lead to cancer tend to affect cell growth and division and death. Right, so if you take normal cells and put them in a petri dish and you add growth factor, then they begin to divide. And if you remove growth factor, then they will stop dividing or grow less. Cancer cells grow uncontrollably. If you put cancer cells in a petri dish, regardless of what else you put in the petri dish, growth factors or not, they're going to divide a lot. So cancer cells, for instance, might create their own growth factors. They might divide regardless of whether growth factors are present. So this is the defining characteristic in some sense of cancer cells, is uncontrolled growth, and they refuse to die from normal means. They have a sort of immortality, really. So this comes from a change in DNA, but as I say, looking at DNA is hard. So in the rest of the talk, we're going to focus indirectly on these changes by looking at proteins. But first, let me show you just a series of steps that could lead to cancer. What do I mean by an accumulation of many small things? So here's a hypothetical series of mutations. all of these are either going to increase the likelihood of division or decrease sort of control and regulations, like cutting the brakes, so to speak. So for instance, you might have a first mutation going from the pink to the red cell that inactivates a regulator. So inactivates something which slows down the cell from dividing. Okay, maybe that's not cancerous yet, it just divides more. Maybe you might have another mutation afterwards that overactivates a positive cell cycle regulator. That will make it divide faster, so now it's getting worse. Maybe now you have another mutation that turns off a stability factor, which protects you from having more mutations, and now the brakes are cut and this thing just gets more and more mutations until it ultimately has something which you would call a cancer cell. So this is the series of steps that happens. They're all changes in DNA, which propagate to changes in the proteins. Okay, good. So if we want to focus on proteins, I might tell you a bit more about what proteins are. Again, if you've had biology, you might know that proteins are effectively a Swiss Army knife, right? You use them for all sorts of things, to make enzymes, for hormones like insulin, which is implicated in diabetes, of course, for antibodies, to carry iron in the blood via hemoglobin, many, many different functions for proteins. And if, like me, you have the misfortune of taking an L-tri-C anatomy and physiology class in your sophomore year, you will be force-fed this line that form fits function, that everything in biology in the body has a very specific shape, which is geared to what it's going to do. As an example of that form fits function, which is not related to cancer, but I think illustrates the point very nicely, this is how muscle contraction works. Right? So in your muscle fibers, you have this actin protein, which is this long spindly blue guy at the top, and this myosin protein, which is this guy at the bottom that acts basically like a motor. And the form or the shape tells you the function. This myosin guy has this lever and a head here, which attaches to the actin. And then when you clench your muscles and contract, this myosin head, this lever, sort of turns to move and exert this kind of pulling on the actin at the top. That pulling spends some energy, which needs to be replenished by ATP. If you've seen enough memes on the internet, you've probably heard lots of stuff about how the mitochondria is the powerhouse of the cell because it makes ATP, whatever. That ATP then puts this little lever back into place and it reattaches. So here's one example of how proteins do a very specific function, and their shape is kind of uniquely evolved to serve that purpose in an efficient way. Good. So this is one protein function, but not the kind that we're going to care about. What we care about for this talk is signaling. So another thing proteins do is send signals within the cell that control growth and replication. So here's a simplified example. It's called signal transduction, which is kind of a whole end-to-end pathway. So for instance, you could have a ligand, which is also a protein. So there's a protein outside the cell, or it could be something else, like, I don't know, it could be not a protein, but some fat or even some ions, like calcium ions that are involved in muscle contractions. So you have some ligand, which is often a protein. It attaches to a receptor, and this causes a sort of cascade of different interactions between proteins in the cell. Right? So this blue thing does something which affects this red one, which does something that affects this other red one, and then something happens. This is kind of a flowchart or a series of events where at every step there's a protein doing something to some other protein. So these are the things that we're going to care about, proteins which do this signaling. Now here's an unsimplified version. You might say, okay, this doesn't seem so hard. Why is this such a challenging problem? Here is a more realistic pathway for signaling within a cell. And you can barely see it because they're so small, but all of these little words down here are different proteins, and oh my god, that's not what I wanted. Oh, I scrolled and I didn't want to scroll. There we go. All of these are different proteins, and all of these arrows represent those interactions that they do by binding and doing something to some other protein. So it's wildly complicated. And sometimes this RAS protein here, look at this guy has 4 different proteins all sort of clustering on him. I almost said a different word there. 4 different proteins that all interact with this one. So you have this very complicated dependency structure that makes it hard to understand, if one step in this pathway changes, what is the result going to be? Because there's so much going on. It's a challenging problem to solve, So if we want to see how these pathways are involved with cancer and metastasis, the first thing we have to do is find a representation, a way of writing down what we mean by this sort of interaction pathway that captures the important data in the thing. So that's what we're going to try to do. That's the end of the Act 1 before an interlude. So the takeaways were that cancer screws up DNA. DNA is used to make protein by the central dogma through RNA. And one of the things proteins do is send signals by complicated pathways. Good. Lots of dense information. So you should yell at me and ask questions now.
Yeah, let you ask questions and give death threats.
Yeah, those two. Thoughts from anyone while I chug Diet Coke?
This is another one of those days where what we're talking about is so beyond my current knowledge that I can't generate questions.
Ah, good. Okay, so can you identify one point where something is confusing or something that doesn't type check? Like, which one of these statements were you not able to parse? Pick one that didn't make sense to you.
All of them made sense individually. It's just that I could not. I don't understand it well enough to ask questions about it.
Okay, that's fine. That's where you should be, by the way. That's not something to be unhappy or embarrassed about. That's the goal.
You see, you're referencing bioclass a whole lot, and like you talked about this in bioclass, and I'm pretty sure I'm like the section before we actually talk about this in bioclass.
Ah, my mistake. Okay. Well, this is a preview then for you. You'll know when you get to this this central dogma stuff. You'll already have some idea that this is a flowchart. But okay.
Yeah.
Oh, random idea. Marginally related, but that mitochondria is the powerhouse of the cell thing has actually saved me a lot of times.
It's such
I'm
a
gonna be
good
honest.
game.
That and the Weird Al song about the pancreas.
Oh, yes. Islets of Langerhans is in there. I was actually singing
Yes.
that. Oh my god, when I took A and P. Jesus.
I'm gonna be honest, you said mitochondria is the powerhouse of the cell, and my brain just got to shut down for a moment.
It's a terrible meme. I apologize for referring to it. I didn't really need to. Okay.
Is there just one quick question? Do you know? Is there a straightforward way to actually observe all of these pathways in a cell? Because drawing this diagram by, I assume there must be some kind of an experimental method for determining how these proteins interact within the cell. But
Are you intentionally trying to preface what I'm about to say next or something?
no, actually, I was just curious.
It's a very good question. Yeah, I sort of pulled.
Did not see your slides, I promise. But
I was going to say, are you working with Putin? You hacked my computer.
no.
Yeah, that's an excellent question. You could say, this has a whole amount of information that you seem to have pulled out of your ass. How do people know what these things interact with? It seems like this would be very challenging to observe, and you're right. So I'll tell you a little bit about how you tell whether a pair of two proteins interact in one of these pathways. It's an extremely laborious process, but here's one way you do it, by this so-called two-hybrid screening. So let me tell you what in the lab you do for this two-hybrid screening. So suppose you want to test, you have two proteins in that flowchart I just showed you, and you want to test, does the first one, protein A, interact with the second one, protein B? So here's a simplified version of what they do. You take a simple organism like yeast that you can grow easily in the lab. Yeast has some DNA, which is this bottom strand, which I'm circling in pink. So yeast has some DNA, and part of that DNA are sort of activator regions. So this UAS is an upstream activation region. So when this upstream activation region is activated, hence the name, you begin transcription. Remember, DNA is transcribed to RNA. So when this thing is activated, this guy goes, it fires, you make this RNA, which eventually leads to protein. This is the lac operon, which is classic AP Bio thing. So anyway, when this thing is activated, you get RNA and then eventually get protein. Okay, fine. How does that help us tell? We have two proteins we want to test if they interact. How does this help us? You take the first one, protein A, which I call the bait. You take the bait and you glue it to the activation region. So now it's blocking the activation region. you're not going to get a lot of this protein. You get a little bit because sometimes, you get transcription which starts here, so you get a very small amount of this mRNA and then the protein. So you get a small amount here when the bait is blocking the upstream activation region. Then what you do is you take protein B, which I call the prey, to see whether these two interact, and you dump protein B into the petri dish. Now, one of two things happen. Either the bait and the prey do not interact. Protein A does not interact with protein B, in which case these things never bind. This thing stays sitting on top of the activation region, and you do not get a lot of protein. Or if they do interact, something else happens. If they do interact, protein B binds to protein A, the prey binds to the bait, which allows activation to happen. If they do bind, then activation, the upstream activating region fires, you get transcription, and a large amount of the reporter gene, as it's called, the reporter gene is transcribed, and you get the associated protein. So this is a fairly straightforward way of doing it. If you want to test whether these two guys interact, you do this setup where you glue one to an activation region, dump in two, and if you get a lot, they interact. If you don't get a lot, they don't interact. So you can imagine kind of roughly taking maybe, tens of thousands of grad student hours, which is the standard unit of work in scientific fields, I think. All you do is take basically the power set of all of these proteins. You basically just take any two, pick any two. So this is like n choose two. Pick this one and this one, glue one to the yeast, or dump in the other one, see if you get a lot of reporter gene, and if yes, they interact. If no, they don't. Then okay, those don't, and then you pick another two. All right, those don't, and then you just keep iterating this process pairwise until you know which ones interact and which ones don't, which is extraordinarily laborious. Many, many studies are published on this.
Christian, this is somewhat of an aside. Are you familiar with the term graduate student descent?
No, what does that mean?
It's a play on a gradient descent, and I don't know. It's not really relevant here because there's no gradients, but I just thought that you would find it funny.
That's really funny. It's like an optimization routine where you want to tell whether something is true, so you just dump a bunch of graduate students on it, and then give them PhDs if they're getting closer to the answer, and kick them out if they're not, and then eventually you converge to the answer. Oh, it's a joke, but that's actually how science works. Oh.
Yeah.
Why am I laughing? Good. Okay, so this is one way of doing it. You can imagine doing this to tell whether these guys interact. I should quickly say there's other ways of doing this. This isn't the only one. I'm actually, some of these other ones I think are very cool. There's some biophysical methods that you basically scatter light off of samples, and you can tell based on the way that the photons bounce off if these guys have bound or not bound. Basically, this is based on understanding of the interface between the two proteins. So there's certain electrostatic forces that cause these things to be attracted at a certain place where they interface, and you can scatter light off of them, and if your apparatus is sensitive enough, you can actually tell whether they bound based on those electrostatic effects. So that's kind of cool. You can use magnetic resonance, lots of other techniques. Since it's expensive to do this in the lab, there's some Foley simulational techniques where you just have a protein folding simulation which gives you a shape for the protein, and you just drop two of them on a computer and let them simulate it and see if they interact. I think that's very cool. There's a lot of ways to do it because it's hard. I say this only because in the paper, the breast cancer paper, they come up with their own, they come up with their own protein-protein network, and they list a couple. Here's where they list it. They say, our pooled data set comes from the yeast 2 hybrid experiments, some predictions from basically people thinking things are supposed to interact, and looking at the literature, which basically means going through this graduate student descent algorithm and looking at what they found for some of them. But anyway, I'm rambling now. But does this answer, Adam, your question about how you tell whether these things are interacting?
Yes, and it makes me feel very sad for all of these students.
Mm-hmm. Man, it must suck to be a bio grad student.
Oh, poor bio. They do important work.
They do. It seems grueling. Good. Okay. If there's no other death threats, this is other. I say other because that was one. This is what I think is the most interesting part. This is why I'm interested in this paper and this on this question. Really, is using graph theory to understand this.
That's familiar.
Yep. Okay, so let me be a bit more precise about what I mean by these interactions, right? So I told you what they were a moment ago, these proteins basically binding, but to be very precise, I'm going to say a protein-protein interaction is any scenario in which one protein makes physical contact with another protein and changes something about it, changes its state. So we've seen some examples. There was the muscle, the myosin attaching to the actin, the enzyme, the classic example from bio, some enzyme binds to a substrate. If they're both proteins, I would call this a protein interaction because they're physically making contact here. The other picture we saw of a ligand attaching to a cell membrane is an example of an interaction. So this is just a rehash of what we were saying about interactions a moment ago. And now you could ask, what do we need to understand about these interactions to answer the breast cancer question? What part of this whole thing is relevant? So understanding all of these interactions is very, very hard. It's a field called protein biophysics, and I can put these textbooks in the Google Drive if you want. I looked at two in making these slides, this mathematical approach to protein biophysics, this protein-protein interactions, very complicated field where you need to model precisely the interface between these proteins. So you need some biophysics, some quantum chemistry, some molecular dynamics. This picture I took from a paper where they used nuclear magnetic resonance, where you're basically putting this whole guy in a magnetic field and then sending in a little pulse that makes all of the nuclei rotate and then send off a return pulse to tell where all the nuclei are and get a picture of how these two proteins are fitting together. It's very, very complicated. So I say all of this not because we need it, but to say that for the breast cancer problem, we can actually throw away all of these details about the way the proteins talk to each other and just focus on whether they interact at all. Just a collection of proteins and which ones talk to which other ones, how they interact. That's very convenient because we can use a well-studied mathematical structure to do that, called a network. So this is the punchline of the talk. This is the tool that we need. Just take every protein, so proteins A, B, C, and D, for instance, represent each of them by a dot or a circle called a node, and if they interact, connect them by edges. So that's it. It's just a bunch of dots connected by edges. This structure is called a graph or a network, and they're ubiquitous. They show up over and over and over in science, social science, mathematics. Surely already you can think of several different places where you could apply this structure. So you could use this to model Facebook, for instance. You could have the dots are people and the edges are friendships, people you're Facebook friends with. You could use this to model computers on a computer network. Many, many, many things. You could use them to, each of these are atoms in a molecule and lines are bonds. Many applications of this. Okay, so this is the structure we'll use, this network. And to get you thinking a little bit, although we won't need this for the paper, I have a question for you to give me a break from talking, so I'll let you think about this and give me an answer. Suppose I have some big old network, or a graph, which as I say is a bunch of dots connected by lines, and someone gives you the following task. You need a way of giving each one of these guys a score or a number, which measures how important or how central that guy is to the graph. So if these are proteins, you want to have a way of assigning how important is each of these dots to the whole network overall. So for instance, I've colored this graph with one way that might suggest what I mean by important. We might say the blue ones are more important or more central to this graph because just by looking at it, it seems like they're somehow more more important to the overall structure of the network, and the red ones are less central. Okay, so think for a moment. There's many answers, and I'll take as many suggestions as you have, but if you wanted to come up with a number to measure how important or how central one of these nodes is in a network, how would you do it, or what number would you use?
So I'm reversing what you did here. It looks like you could define your centrality using how many bonds
You're such a
does... sure.
Sorry, go on.
I'm just find all the bonds around a given atom. God. Find all the bonds around a given node and count how many nodes are. How many second level nodes are there? I don't know how to describe it any differently.
So you're saying count how many nodes can be reached by a two-step path from the existing one, right? Like this guy can go here, then here, here, then here. You're going to count. That's your measure of centrality.
Yeah.
Good. Okay, so count how many nodes can be reached in 2 steps. Interesting. Okay. Other suggestions?
I want to say you could possibly like determine centrality given how often or how many nodes are grouped together. Like, how do I state this? I guess the more nodes grouped in an area would dictate how important it is or its centrality, right?
I see. So you're looking for clusters like this guy, for instance. This guy that's like there's a bunch of nodes that are all highly connected to each other, in some sense.
Correct, and yes, correct.
Okay. Yeah. So look for, I guess, highly connected. I guess I'll call them subnetworks. So highly connected subnetworks. Yeah, and you would say that people or nodes in those highly connected subnetworks are somehow central. They're the important ones.
Yes.
Good. Other metrics.
So, I'm going to draw on prior experience.
What are you going to propose? Log prime?
Actually, no, never mind. I was thinking
Stop it.
about something we've done.
What were you going to say?
Oh my god, I totally missed that.
Bad joke.
Okay, yeah, the thing I was going to say would be concerned about the graph as a whole.
What was the thing you were going to say, though? It might still be relevant.
I was going to bring up the chemical graph theory
Yeah, but
and
what metric?
take the eigenvalue of the connected nodes as a matrix.
You want to take the largest eigenvalue of the adjacency matrix? Is that what you're saying?
Yeah, but that only applies to the entire graph.
Yeah, but how could you... Okay, so you have an eigenvalue... everyone's going to be totally lost now. How can you take... You have an eigenvalue. You have one number lambda associated with the whole graph. And what other object comes along with an eigenvalue that would give you a different number for each node? What else could you use? These things come in pairs, right? What comes along with an eigenvalue?
An eigenvector, but how does that help us?
How long is the eigenvector here? Like, what is its dimension? If this thing has n nodes.
It's 1 by n?
Yeah, so the adjacency matrix is n by n. This eigenvector is 1 or n by 1, right? So this guy has one number, if I call them v1, v2, up to vn. This guy has n numbers, and each one of these numbers is associated with one of the nodes, right? Vn is up to the nth node. So do you see a way of assigning a centrality score using the entries in this eigenvector that gives each one of these guys a score?
Would you just go through the vectors and color them in order of eigenvalue?
Yeah, you just go through, you go through the entries in this one vector, right? You pick the largest eigenvalue, go through the entries in this vector, and then just say this guy is the score for node 1, this guy is the score for node 2, this guy's the score for node n. You just assign to each node a score, which is his entry in the eigenvector, like you were saying. Good. That's an incredibly insightful For the other people in the audience, I did not tell him to answer that. So that's a very clever way of measuring centrality, which appears on the next slide. What you just defined without being prompted is something called eigenvector centrality. That's actually if the circles in your network are websites and links between them, and edges between them are links between websites. You might ask, how do I determine which websites are the most important, the most central, the ones that should show up on my Google search, right? The way that Google measures the centrality of websites uses not exactly, a small change, but the same idea that you just told me, eigenvector centrality. Take the largest eigenvalue, find the eigenvector, assign guys their scores from that. Okay, I shouldn't spend too much more time talking about this because there's maybe two people that understand what I'm saying. So, but I'm very impressed that you came up with that because that's basically what Google does, and it's a very powerful thing. But okay, good. So the other suggestions that we had were so these ideas of so degree centrality. This is just counting how many edges you can reach with a path of length 1. Logan proposed a way of counting how many you can reach with a path of length 2, which is slightly different, but related to degree centrality. These closeness and betweenness centralities are related to the idea of looking for highly connected subnetworks, right? Because these things have to do with not just an individual node, but kind of subnetworks of nodes, and in particular, paths between nodes. So for instance, closeness centrality just says find the average length of the path that you need to get between one node and a second node. Average that over the whole graph. So guys in highly connected subnetworks, as in the second suggestion, will have higher closeness centrality. Good. So you guys basically got close analogs of all of these in your suggestions. Good. Okay. Thoughts on that was a fun exercise. Thoughts on centrality before moving onward.
How is this at all related?
Good. Yeah, it's really, for the paper itself, this is less related. This is very important in graph theory generally, and if you wanted to understand for protein networks, which of the proteins that would be, say, most damaging if you remove that protein from the network, which ones are most relied upon metabolically. If you wanted to answer that question, you would use centrality and look for the highly central proteins. So I included this mostly just for spiritual well-being, to get you thinking about graph theory and networks. But as I say at the bottom, we won't need centrality or any of this for this paper, but this was an example to make you think about graphs and networks. Good. Okay. One more brief aside and then back to the paper, and we'll be very serious. So if you are interested in this graph theory or network theory stuff and you have Python installed on your computer, you can play with networks in a nice, clever way using this library, this NetworkX library. So it's very easy. It's all built in. You just make a graph using the syntax. Here I've imported this NetworkX library as NX. So you just do nx.graph. You can add 10 nodes by using range from 1 to 10. I just put in by hand some edges. So here I've just put 1, 2, means connect guy 1 to guy 2. And I connected 1 to everybody. Then maybe I just added a few more random ones, and then I just draw it. So here's what it looks like. So if you want to play around with graphs, you could do this and just make your own like this, add in some edges. And all of these numbers I was just telling you about, this will compute for you. So you can do in Python, you can just do closeness centrality or eigenvector centrality and just use these words and it will compute the numbers for you. And you can just print out what all of them are. So you don't have to do any work, it just knows how to do those under the hood as built-ins. Good, so that's all on the side. Now back to what we actually care about. So that was all an aside on graph theory, to convince you that graph theory is cool. But back to the paper. So again, we're thinking about breast cancer, and we want to answer the question of which subnetworks, so these are smaller connected parts of a larger graph, which subnetworks are related to metastasis of breast cancer. So when we come back to the final act, I'll just return to the paper and tell you how they use some statistics to find the subnetworks that have to do with breast cancer metastasis. That's the answer we're trying to do. And this big graph is sometimes called the protein interactome. This word is supposed to make you think of the genome. The genome is all your genes. The interactome is all the proteins and how they interact. Okay, that is the end of the graph theory part. I have an interlude before the finale, so thoughts or objections about all this graph theory stuff before that?
So how did you color your colored graph?
How did I color the... Oh, good question. This is betweenness centrality. So this is the third one, sorry. This is...
Super useful in transportation networks. Sorry.
Oh, that makes... So is this something that people would like, if Amazon had a bunch of nodes that were like their delivery centers or something?
Yep, it's very important in studying traffic networks. Betweenness and closeness are probably the two most important heuristics for those.
Okay, so people that do like logistics or what's that word called for?
Like
What's the field called when people like optimize shipping and stuff?
supply chain. Well, there's.
Yeah,
I guess it's kind of a field of supply chain management. I guess.
That's what I was thinking of. Supply chain management. Good. Okay. Almost done. Let me tell you a bit about p-values. So one of the things they use in this paper, and if you choose to go back and read it afterwards, they report something which is very common in the biological and social sciences, p-values for things. So let me tell you what a p-value is. So say you run some experiment and you get some answer, some result, which is a number, an outcome. And you want to know, was that outcome due to what I did, the treatment, or could I have gotten that outcome by pure chance? So the p-value is just the probability that if I got a data point here, if I got this number, what is the probability that I would have gotten a result at least that good, so that large or larger, from pure chance, from without doing whatever I applied to the experiment, but just randomly. So it's kind of the probability that I could have fooled myself, right? That that my experiment is a false positive, that this could have just been pure chance. That's very abstract, so let me give you a specific example. Suppose you have some friend who's a bit strange and he thinks he's telekinetic and he believes that he can change the result of a coin flip with his mind. So you might have the hypothesis that if your friend uses a telekinesis to try to make a coin land heads up, it will land heads up more often than usual. So you run an experiment where you flip the coin 20 times and your friend is being telekinetic at it, and you get 14 heads and 6 tails. And your friend sounds very pleased with himself, and he says, look, I got it more than half and half. You know, it's more than 10 and 10. I got 14 heads with my telekinesis. And you want to know, what is the probability that this is actually due to an effect, that it wasn't pure chance? So to find that, you could just run a simulation. So I'll go back to Python, but even if you don't know Python, you can probably just tell, this is almost like pseudocode, you can tell what it's doing. I'm just going to do, say, 10,000 experiments. Each experiment I'm going to flip a coin 20 times, and then I start a counter for how many times we get at least 14 heads. And then in each experiment, so for experiment in range number experiments, for each of those 10,000 experiments, I start a counter, no heads. Then for coin flip in range, the coin flips per experiment, so for 20 times, I generate a random number that's 0 or 1. It's supposed to be tails or heads. And if the random number was 1, I increase the number of heads in that experiment. Then at the end of the experiment, if I got greater than or equal to 14 heads, I add 1 to my counter of positive results. Right, so this is just running 10,000 experiments. In each experiment, I flip a coin 20 times, and I count in how many of those 10,000 experiments I got at least 14 heads. Then at the end, I print it out, and it turns out that the answer is about 5%. So 5% of the time with no telekinesis, this would have happened anyway. That's the p-value. 5% of the time with no telekinesis, you could have gotten at least 14 heads. I'm going to flash a slide and then go right past it, but we didn't need to simulate because there's a formula for this, which is known as the binomial distribution, and I could have gotten the correct answer with some math. But don't worry about that. A final comment before the conclusion. This is a bit of a source of perhaps inefficiency in science, because people will usually only publish their paper if they get a low p-value, p less than 0.05. So there's a strong incentive to reach this p less than 0.05, which means that one might be tempted to misuse data or analyze it in such a way that makes your p-value smaller so that you can publish and get sort of the career benefits from your science. So there's a word for this. Misusing data to reach this threshold, this p-value, is called p-hacking. So it's entirely possible to have a small p-value, but still a result which is not really meaningful or scientifically valid. Here's a classic plot from Spurious Correlations. This plot shows in black the number of people killed by venomous spiders each year, and in red the number of letters in the word which won the National Spelling Bee. And they seem highly correlated. So if you computed the p-value, what is the probability that you would have gotten two sets of random numbers that follow each other this nicely by pure chance? You would get p less than 0.05. But that doesn't mean that these things actually cause each other. Clearly, the number of people killed by venomous spiders has nothing to do with the spelling bee. So it's not always true that p less than 0.05 implies, a good verifiable scientific result, right? This is the point I'm trying to make. Good, a lesson to take home. All right, last thing, in the remaining slides, I'm just going to go through the paper again with the new things that we've learned and tell you what they did, and then we can wrap up. So thoughts on p-values or p-hacking before the conclusion?
Negative results are results too.
Yeah, but there's no incentive to publish negative results.
Yes, which is a failure of modern science. But
yes. I mean, this is why I do physics, where there's no p-values. I mean, you either have a proof or you don't.
Context necessary.
Yes. Is someone posting bad quotes from me? Dot quote ads, so no heads. Good. Yeah, that's interesting. Okay, let's go back to the paper. So at the very beginning, I showed you a flowchart from their paper, but it wasn't actually the flowchart from their paper. I took the flowchart, which was on page 3 of their paper, and I simplified it by removing some of the complicated stuff. So now I'm going to show you the whole thing end to end in full glory. So again, what did they do? They started with a protein-protein network that they got from yeast and from curating stuff from the literature, and a bunch of samples of proteins from people that have breast cancer, but some have metastasized and some haven't. Good. Then they fed them all into this table. I'm trying to scroll down and it's not working. Oh, because I have the pen on. They fed them into this table, which I have down here, and they computed some statistics. I'll tell you about those in a minute, but what they're doing effectively is for each protein, they're putting a number in this table that tells you the amount of expression, so the level of that protein in that patient. So all of these are measurements of protein expression. Put a bunch of numbers into this table, then you look at subnetworks in your protein network, and for each subnetwork, a subnetwork has many proteins, which are many rows. So say for this subnetwork, we look at the first row, the second row, the third row, the fourth row, the fifth row, because those are the 5 proteins in this network. And we combine those 5 rows into a single row in a way to get a particular sort of weighted average that tells you for this subnetwork, these are the measurements of those proteins averaged in a certain way across the different patients. Now we just want to find, are those numbers, is the measurement associated with this subnetwork related to metastasis? So to find whether these numbers are related to metastasis, they use something called mutual information, which I'll tell you about briefly. But let's skip past that for a moment. I just want to tell you the answer, the punchline they get. By picking the subnetworks that have mutual information with metastasis, they pick out a few special good ones. So these are subnetworks that sort of maximize this mutual information, have the largest amount of data about metastasis, and then they use these for their prediction. They say, I'm going to look at these subnetworks to predict whether the next person with breast cancer has metastasized or not. That's what they did. So I have those in the slides chopped into pieces, but I guess I can skip through them. This was the first part, the second part, and then the third part. So all I need to do is basically describe to you this statistical or mutual information thing they did. So I'll flash a formula, although you don't really need to understand it. What they were doing basically is trying to find whether the numbers for a given subnetwork contain some information about metastasis. And to do this, they use this tool called mutual information, which comes from information theory. There's a formula for it that involves summing up some probabilities times some logarithms of these ratios of probabilities. But the formula is less important. You can think about this as sort of a Venn diagram where h of y is the amount of information, or if you studied chemistry, h is actually an entropy, not unlike, well, there it's the Boltzmann entropy, but here it's the Shannon entropy. It's not a big deal, but you can think about the amount of information in, say, the metastasis statistics, so whether they metastasize or not, and one of these other Venn diagrams is the information in the subnet measurements, so the amounts of proteins in the subnetwork measurements. And then you're just asking, what is the overlap? How much information do I get about the other one by measuring the first one, right? That's what mutual information is. I mean, this is very abstract, so let me just give you a concrete example in Python. I'm going to do a simulation. I'm going to roll some dice. I run, again, 10,000 trials. On each trial, I roll a die. I get a number from 1 to 6. Then I roll another die, get a separate number from 1 to 6. And then I compute whether the first roll is an even number or an odd number. So this is going to be a 1 if it's even, and 0 if it's odd. Then I just put them all into a list. So at the end of this experiment, I have 10,000 first dice rolls, 10,000 second dice rolls, and then 10,000 numbers that tell me whether the first dice roll is even or odd. And then I just print out the mutual information between the 2 dice rolls, and then between the first dice roll and whether it's even or odd. So mutual information is always going to be between 0 and 1, by the way. So as you might expect, the mutual information between these 2 totally unrelated dice rolls is very close to 0. Knowing the first dice roll tells you almost nothing about the second dice roll. But the mutual information between the first dice roll and whether the first dice roll was even or odd, this is quite large. This is about 0.7. So knowing something about one of these 2 things tells you quite a lot about the other one. Clearly knowing the number tells you all of whether it's even or odd. It's not 1 because you can't go the other way. Knowing whether it's even or odd doesn't tell you the whole number. So that's mutual information. Good. So what do they get? At the end of the day, by using this mutual information, you pick the subnetworks that have small p-values in the size of the mutual information. So it's very unlikely that you would have gotten that high of a mutual information by chance. There's this word here about greedily choosing subnetworks. What do I mean greedily? Well, what if one protein is in several different good subnetworks, right? It would be, you want this guy, you don't know whether to assign this guy to one or the other. By greedily, I mean you just start with the one that has the highest mutual information, and then you put him in that one, and not in the other one. So you break ties by putting the guy into the one with the higher mutual information, and then you go downwards. That's greedily choosing. Good, so at the end of the day, this is the answer they get. They get a bunch of subnetworks that have high mutual information with cancer metastasis. Now here they're labeled with some colors to tell you whether in the cancer patients there is more or less of that gene and therefore that protein. So the red ones are upregulated, and these guys, the upregulated ones, tend to be related to CTSNR, which over here are signaling cell growth, signaling cell proliferation. So you upregulate the genes that signal cell growth. And these green networks they find, green ones are downregulated, and these green ones seem to be related more to other CT remodeling, S and CR, I guess cell proliferation. So these ones, I guess these would downregulate control of cell proliferation. Either way, upregulating or downregulating, these proteins are associated with more metastasis. Good, that's what they find. And then the rest of that paper are effectively the performance metrics, the numbers that I told you before. So by using this way of picking networks, they get better prediction accuracy, the 72% beating the 63% benchmark. they find that there's more consistency between patients. Subnetworks that are associated with metastasis tend to occur over and over between metastatic patients rather than in just one with a small p-value, so that's significant. And perhaps one of the reasons why this paper was cited so much is that this thing sort of singles out new directions for research, because it tells you these are the protein pathways that are important for cancer research, so focus on understanding those more, right? This is a way of generating new hypotheses. Good, good. So that's everything. The TLDR is that we used networks to capture the protein data, and that gave us more power in predictions. Cool. Now death threats. There should be a lot of death threats now.
So, did I understand it correctly that you're able to observe the presence of a protein, but not necessarily the entire structure of a given subnetwork that they're pulling out?
You're able.
And they're.
So you're talking about back the things that they're feeding in are observations of proteins and not
Yes.
the networks? Yeah, exactly. So
Okay.
yeah. So you can't measure directly the activity of an entire network. So as a proxy, they say the activity of this network is going to be some weighted sum of the activities of the individual proteins in the network. Yeah. Good. More thoughts?
The concepts are clear enough.
You don't sound happy about it, though.
It's interesting. Why would I be happy to be studying cancer?
That's fair. I mean, I think it was a fun paper. It's not as interesting as centrality, actually, but no, they get some nice results. Good. More thoughts or objections? Or I guess ratings of interest. So how interesting or useful is this, or what changes would you make or suggestions for the next several talks?
It began with medium-low interest and ascended towards the end. Ascended, word.
Where did we, if it began medium-low, did it end high or medium-high or just medium?
Medium-high.
Okay, good.
Too high.
One of those Dr. Face charts.
Oh yeah, with the amount of pain, like the. Good. Okay, other feedback or things you liked or didn't like